Well, today we have the honor to introduce you to Carlos Martí, who holds a PhD in bioinformatics. He studied a bachelor's degree in biotechnology at Polytechnic University of Valencia, where he had his first encounter with bioinformatics, studying the assembly of the genome of the courgette. After that, he studied a master's degree in bioinformatics at the Autonomous University of Barcelona, which led him to pursue his PhD at the Spanish National Center for Cardiovascular Research, studying alternative splicing events from RNA-seq data and their implications in cardiovascular disease. So today, Carlos will talk about how Bayesian statistics can help with the characterization of alternative splicing evolution, showing us how models of evolution of quantitative traits can help us better understand the contribution of alternative splicing to transcriptome diversity. This sounds like a very interesting topic for us bioinformaticians. So thank you, Carlos, for being here today. And without further ado, I'll let you begin with your talk so you can share your screen whenever you want.
Thank you, Marisol, for the introduction. Let me see. Do you see my screen now? Can you see my screen?
I think so. Yes, I can see it, but
okay, so
I think that the people in the streaming can too.
Okay, so thank you all for coming to this new edition of this bioinformatics seminar series. And today, as Marisol said, I will speak mainly about how quantitative modeling of alternative splicing evolution can help us to better understand how this process evolves, really. So I will start with a brief introduction in which all of us more or less know that eukaryotic genes are often divided into introns and exons, such that when the gene is transcribed into a primary transcript, there are some splicing factors in the nucleus that recognize some signals in the primary transcript and assemble around them to catalyze the splicing reaction. That consists in the removal of the introns and joining together the exons. This can happen in more than one different ways, leading to the production of different possible transcripts from the same gene. And this transcript may be produced in different proportions. Some of them may be considered to be aberrant transcript, and after transported to the cytoplasm may be recognized by the non-cell mediated decay machinery and degraded. The combination of the non-cell mediated decay and alternative splicing provides an interesting mechanism for gene regulation that is very well known to have a functional impact in some cases. One of these examples, we can find it in the sex determination system in Drosophila, which is initiated by the inclusion or skipping of an exon with an in-frame stop codon, which upon inclusion targets the RNA for degradation. Moreover, as a result of degradation and production of these different transcripts, we end up having steady-state RNA levels in which we have different transcripts produced by a single gene in different proportions, which is what we usually represent by this Greek letter psi, and we also call PSI, stands for percent splicing. Now, these transcripts that are now in the cell, some of them may be translated into different proteins and potentially code for different functional proteins. This is the case of the IgM gene, which was one of the first characterized to be alternatively spliced, and which allows it to produce different functional proteins, one that is secreted and one that is bound to the membrane. However, it was not until a couple of years ago that we started to know how frequent this was at the whole genome level. First studies with RNA-seq showed that over 95% of human multiexonic genes actually undergo alternative splicing. However, we were interested in seeing whether these transcripts were translated and producing different proteins, which was the role that was first envisioned when introns were discovered. So with ribosome profiling experiments, what they show is that most of these, at least exon skipping events, are also bound by the ribosome, suggesting that the products of this alternative splicing are often translated. However, when it comes to detecting directly the protein with mass spectrometry-based proteomics, they only found that 2% of human genes are actually producing different proteins. Despite the technical limitations that proteomics has, this seems like a very big gap and raises the question about the general functionality of alternative splicing. An alternative approach to try to understand this problem is by studying the evolutionary patterns of alternative splicing. First comparative studies in 2012, they took RNA-seq data from different species and different tissues, and they identified some exons that were alternatively spliced across the different species. Here we show a classification in which, in capital letters, they show the type of exon that is. So in capital letter is alternatively spliced, and in lowercase we have whether they are not alternatively spliced. So what they showed in this study is that over about 2,500 exons that are alternatively spliced in mice, only about 500 of them are alternatively spliced consistently across all 4 mammalian species, suggesting that only about 20% of alternatively spliced exons in a single species are also alternatively spliced in different species. Similar results were found in this other study, in which what they did here was to analyze how many of the exons were alternatively spliced in common with human. And what we can see here first is that for chimp, for instance, that is very closely related with human, we only find that 55% of the exons are alternatively spliced in both species, suggesting that concentration of the alternative splicing is relatively low. However, this is a rather qualitative definition of alternative splicing, either yes or no, and we are often more interested in the quantitative level, right? So going back to this slide in which I mentioned when this proportion, we are more interested into studying how these differential splicing rates are evolving over time. So in these studies also, what they performed is they took these estimates of exon inclusion rates across the whole transcriptome and compared them in a pairwise manner across all samples analyzed, in which they included different species and different tissues. And in this plot, they show the correlation across all possible samples, and in different colors here on the rows, we have the different tissues. And what they show in this type of analysis is that the samples corresponding to the same species tend to cluster together over those samples belonging to different tissue, to the same tissue, suggesting overall that alternative splicing may contribute to explain the differences between species. However, this is something that is rather difficult to interpret. So for instance, we don't know really whether 20% conservation across four species is high or low, or 55 for human and chimp. And this species driven clustering, we don't know whether it is really expected or unexpected and under which scenario. So despite these observations being interesting per se, we try to address this question in a different manner. And we try to ask this other type of question, such as what is the relative contribution of different evolutionary forces to the evolution of alternative splicing. So with evolutionary forces, here we will be talking about random forces, which are mutation and drift, and natural selection. For instance, when we are analyzing sequence, we usually are not that interested into the similarity of different sequences from different species when we are trying to study natural selection, but rather on the rate of substitution of non-synonymous and synonymous sites. So ideally, we would try to do something similar in this case. Besides these additional questions, we have some other questions that have not been addressed so far and that we think we might be able to better understand. Like, for instance, do different exons evolve similarly? Are exons selected to be alternatively spliced, or is it something more random? And also that something that is interesting, and we will try to address also in this work, is how much of the alternative splicing changes that we observe between different species are actually selected to be different, or it's just a matter of random noise. So to try to answer these questions, what we propose in this work is to use models of evolution of quantitative traits and adapt them to analyze alternative splicing data. So moving to the results part, what we did here is to collect public data sets in which from RNA-seq data in which we try to collect as many species as possible to try to have a very dense phylogenetic representation of the mammalian species. Here we are only using liver samples for simplicity, also because it's a tissue that is easy to sample and identify in different species, and it's relatively homogeneous and simple for a first approximation. So, we are going to talk about models of quantitative trait evolution, and the first one and more simple is the Brownian motion. With this model, which I will explain briefly afterwards, we would aim to try to understand whether exons evolve in a similar manner. Some exons evolve faster or slower than others. So under this model, what we expect is for any trait, a generic trait that we can call X, that in a short period of time, there is some random variation that is accumulated constantly with time. So well, here I'm representing the independent evolution of different lineages starting from a common ancestor. And we can see that over time, these different lineages accumulate random variation. And after a determined period of time, we end up with a distribution that is centered at the ancestral value and with a variance that is proportional to the rate of evolution, which is what is interesting to us. Thus, if we have different traits, for instance, different exons that are evolving under different evolutionary rates, we would expect that this distribution may be narrower or wider. And we can use this information to actually infer the evolutionary rate for each exon independently. Now, we don't have lineages that are evolving independently, but we have something that resembles more a phylogenetic tree like this. So to try to analyze this data, we can follow a different approach in which we more or less know the trait value at each node of the tree. In this case, I made a simple example here for simplification, and we split the tree in different branches. Therefore, we can calculate the change in the trait for each of the branch, and we know the length of the branch. So we can make a plot like this in which we have time against divergence for the traits. Therefore, what we expect under this model is that the divergence increase linearly with time. And the slope of this line is what represents the average evolutionary rate. So doing this kind of analysis, we were able to estimate the average evolutionary rate for each exon, which is represented here in this histogram. Here it's represented on the log scale. And what we can see is that there is a lot of variation from -1 to 1. There are about 2 orders of magnitude, suggesting that there are some exons that are evolving 100 times faster than others. To better exemplify what these values actually mean, I made some simulations for an exon starting with an inclusion rate of about 1 and letting it evolve over some time. So for those exons that are evolving relatively slow, that are contained in our data set, we would expect something like this, in which the inclusion rate would barely change in hundreds of million years. However, most of the exons more or less resemble this situation in which after 25, 40 million years, we start to see that they start to be very different. While there are some exons that are evolving much faster, and in a very short period of time, they will resemble very little the ancestral value. Overall, what this suggests is that there is widespread variation in the average evolutionary rates across different exons in the genome. Next, we will use this other model, which is the Ornstein-Uhlenbeck model, which is a generalization of the previous one, to try to address the question of whether stabilizing selection is constraining the evolution of exon inclusion rates. So to explain a bit the Ornstein-Uhlenbeck model, this, as I mentioned, is a generalization. So besides this term that represents the random accumulation of variance over time, we have these additional terms which has 2 parameters, mu, which represents an optimal value that maximizes the fitness of the species. So those individuals that carry a value that is closer to this value will survive more and the population will tend to evolve towards this value. Then we have also this other parameter, alpha, which is the selection strength. This is the strength with which the population is pulled toward its optimal value. This can be represented inversely by the phylogenetic half-life, which is the time that is required to reduce the distance to the optimal value by half. So for instance, in this example, we see that the trait besides accumulating random variation, is also deterministically drifting towards this optimal value, which is represented by the dotted line. Therefore, if we have different exons that evolve under different selective strength, we would observe something like this, in which we see that now this other exon with a stronger selective strength drifts much faster towards this optimal value. And this is the type of information that we will leverage to try to estimate the selective strength for alternative splicing. Going back to this toy example, now instead of a linear dependency between the time and the divergence, we would expect that this line starts to saturate with time up to a point in which the variation that is created by the random forces is eliminated by selection. Therefore, the pace at which this line saturates will also give us an idea about the strength of selection in this case. So we did that for our data, and we estimated a phylogenetic half-life of about 25 million years. This value is similar to that previously estimated for gene expression, suggesting that at least selection is as strong for alternative splicing rate as it is for gene expression levels, which in general is relatively low but significant. To better understand the implications of this specific value, again, I made some simulations in which now I took an exon with an ancestral value of 0.5, but an optimal inclusion rate of about 1. Therefore, we can see that the average trait is goes rapidly towards 1, but there are always some exons that remain included at a low rate, even if the optimal value is still 1, suggesting that selection is not sufficiently efficient to remove those exons that are skipped at a relatively low rate, but remain there. Overall, what we see from this type of analysis is that exon inclusion rates are also evolving under stabilizing selection. Next, a natural question that arises from this type of analysis is whether natural selection is driving these heterogeneous rates of evolution across exons. Right, so to try to answer this question, we took the whole set of exons in our data set and we separated them into different groups according to the rate of evolution that we inferred with the Brownian motion model, as shown here in the x-axis. Now, on the y-axis, I'm showing you the inferred phylogenetic half-life. And what we see is that, contrary to what we could expect, the phylogenetic half-life does not increase with the evolutionary rate, suggesting that selection is not driving these heterogeneous rates of evolution. In contrast, what we found is that when we plot the rate of neutral evolution against this average rate of evolution, we find a very clear correlation, suggesting that the variability in evolutionary rates is not driven by selection, but by variability in the rate of neutral evolution. Next, we mentioned in the Ornstein-Uhlenbeck process, we also had an optimal value. In this case, it will be an optimal inclusion rate. These values, what would allow us is to try to study the general functionality of alternative splicing, because we will try to see the optimal values for each exon and then infer whether the skipping of an exon is selected to be that way, or it's just an error because selection is not sufficiently efficient to remove them. So here in this graph, I'm showing you the cumulative distribution for the optimal inclusion rates across the whole sets of exons that we included in our analysis on purple, and also the minimal optimal inclusion rate across the exons for each gene. So from this type of analysis, what we see is that there are only about 2% of exons that have an optimal inclusion rate below 0.9. There are few exons that are selected to be spliced at low rates. And this corresponds to about 5% of the genes in our dataset. To try to see whether this definition of alternative splicing as functional when they have an optimal inclusion rate below 0.9 is relatively meaningful, we took data from BuzDB, which is a database that collects annotated functions for different exons that have been experimentally tested to have different functions. And when we cross this data with our data set, what we see is that those exons that have an optimal inclusion rate below 0.9 are strongly enriched in exons that have an annotated function, suggesting that our analysis makes some kind of sense, and that overall, alternative splicing may be functional only in about 5% of the mammalian genes, and not a general term, and not a general thing across the whole transcriptome. Finally, something that we were also interested into seeing is whether there were changes in the optimal value across exon inclusion rates. This way, we could see whether we should be able to see whether changes across different species are actually driven by selection, or it's just random noise. This is possible because in this type of a scenario in which we have two groups of species that diverge at some point and since then are evolving under different regimes with different optima, we would expect that this group of species here in black try to converge to a different distribution compared with those species that in gray that try to converge to another distribution. In contrast, if the two groups of species are evolving under the same optima, they will converge to the same distribution. So first, before going to our data directly, what we did is to make some simulations in which we simulated changes in the optimal inclusion rate across the phylogenetic tree in our dataset, and we tried to infer them with our model. And what we see is that the correlation is very poor, suggesting that we are not doing a good job with our model when inferring this. To try to investigate why this may be happening, again, I made some simulations in which I took an exon evolving with an optimal inclusion rate of about 1, and suddenly evolving a new optima of about 0.5. These exons that are represented now in a lighter color are virtually undistinguishable from the others, suggesting that there is a lot of variation and that these exons are not distinguishable by eye or even by the computer. However, this was done with a rate of neutral evolution that we inferred for our data. And if we artificially reduce this rate of neutral evolution, we start to see that these two groups of exons would evolve differently and probably are better distinguishable. So again, we made some simulations under varying rates of neutral evolution and calculated the correlation coefficient between the real and the inferred ones. And we see that this correlation coefficient starts to get better when this rate of neutral evolution is lower. Overall, suggesting that in the regime in which we are working with our data, which is around this value, this rate of neutral evolution is too high and makes the changes undistinguishable from adaptive changes. So to conclude, finally, coming back to our main diagram, in which we have a summary of the things that we were able to study with these models. We have seen that there is a lot of variation in the rate of evolution across different exons, that exon inclusion rates are evolving under weak stabilizing selection that leads to inefficient inclusion rates of exons. And then we also saw that this variability in the rates of evolution is not driven by natural selection, but variability in the rates of neutral evolution. We also found that optimal inclusion rates are low only in few cases, which account for about 5% of the genes, at least in the dataset that we analyzed. And finally, when we try to address whether the proportion of alternative splicing changes across a species that were actually adaptive, the answer is that we cannot know yet. So this proportion remains unknown. And finally, I would like to finish by acknowledging the people in my lab, the people in the bioinformatics unit at CENIC, with whom I've been working closely, and also Klaus Vogel from Veterinary Medicine University in Vienna, who has helped me a lot throughout this project. And of course, I want to thank you all for your attention, and I'm happy to take questions now.
Well, so Thank you, Carlos, for this wonderful presentation. Now to the questions. But before, I would like to briefly talk about our association. So let me share my screen. Mm-hmm. So, well, so we are BioInformaticsGRX, and we work to expand the bioinformatics field, especially in Granada, Spain. But now we have a broader audience thanks to these online activities. And we organize talks and workshops to share from the most basic to more expert pieces of bioinformatic knowledge, such as today's talk that was in the expert level. And well, we encourage you to follow us on social media if you don't want to miss any of our activities. And here you have all our directions here, and also on in the description box below of this video. And well, now without further ado, we shall proceed to the questions. So let's see if we have some questions for Carlos. So yes, it seems that Álvaro Torres says, hi, Carlos. What are the limitations of these models?
Well, for instance, in this case, these are genetic models for trait evolution, so they are not particularly adapted to alternative splicing or any other models. So, for instance, they are not taking into account the genetic architecture of the different traits. For instance, how mutations affect the inclusion rates. This is something that we are not taking into account. Also, here, for instance, we have assumed that the exons are evolving independently from each other. This may not be the case because there are, for instance, transregulators that may be changing and affecting different exons simultaneously, and this would, of course, violate this independency assumption. And well, there are a lot of limitations, but this is the best we could do with these models. And I think we could get meaningful and interesting information that was hidden in the data and that it was not shown before in these previous studies.
Mm-hmm. Well, so Anna says, hey, great talk, thanks. How different exons accumulate random variation at such different rates?
How? Well, we don't really know. What the only thing we have seen with this type of analysis is that it's not because some of them are under stronger selective pressure. They seem to be under similar selective pressure, but there is something in the, I would say, probably in the genetic architecture that makes these exons more sensitive to mutations. So even if the mutation rate is similar across these exons, there are some properties that we don't know that make the probably maybe regulatory redundancy or something that makes that this mutation affects less some exons than others. But this is just an speculation. We don't really know.
Mm-hmm. So Jesus Villegas says, hi, nice talk. Could these models be applied to other type of genomic data? If your answer is yes, what type of data?
Okay, so as I more or less mentioned in a previous question, these are more or less generic models that can be applied to evolution of any trait. In principle, these traits are expected to be normally distributed. And this was not the case for exon inclusion rates, which are bounded between 0 and 1, but there are some mathematical tricks that we can do to try to fix this dependency, and we can use different distributions. For instance, here I use the binomial distributions because you either have inclusion or skipping. And in general, they can be applied to, as long as you can find a transformation that allows your data to fit this kind of assumptions, you can work with them. For instance, for gene expression, instead of being bounded between 0 and 1, is bounded between 0 and infinity. So you usually do the log transformation and then assume that this log transformation is normally distributed, and then you can apply already these models.
Mm-hmm. Erika says, hi, Carlos. Why did you make groups of exons for applying OU models instead of estimating the parameters for each exon independently?
Okay, this is a good question. We made groups basically because for a single exon, there is not enough information to disentangle these parameters that contribute to the evolution of the different exons. For the Brownian motion model, there is, because we have like a lot of species and it's only a single parameter. For the other model, it's much more complex, and then we need to have information for many exons. This is similar to when we are studying sequence evolution, for instance. We never expect to have a phylogenetic tree from a single nucleotide, right? So what you do is you pull information from many nucleotides at the same time and assume that they are evolving under the same process. So we are doing some more or less the same here. We needed to have, I'm doing some simulations. I saw that we need to have about 500 exons, 200 or 500 exons to be able to reliably infer these parameters. So that's why we had to do this grouping strategy instead of going exon by exon.
Mm-hmm. So we have another question. Navid says, hi, Carlos. First of all, thank you. What troubleshooting do you use to solve the limitation of the design of these models?
Can you read it again? Sorry.
What is the troubleshooting that you use to solve the limitation of the design of these models? So how do you test these models to see if they are working well or adjusting to reality?
Okay, well, for me, the key thing is to check. I always do with simulations. So for any scenario that I think may be problematic, I simulate data, and then try to see how the model behaves under these scenarios. Then with the real data, it's difficult to know. Something that I did at some point is to, for instance, remove some species from the analysis and then try to predict the inclusion rates for this species and see how well we can predict these inclusion rates from closely related species. But I don't think there is much more that we can do with real data, because we don't really have access to the average across each species. We have only few samples from each species. So basically, we try to focus mainly on the underlying process rather than on specifics of values for each exon or anything.
Okay, it seems that was the last question. I don't know if anyone wants to ask something else to Carlos. Or Carlos, if you want to share something else with us.
No, I don't know.
Well, so
I will just maybe say that for anyone working with comparative data sets, there are packages also to work with this type of models, and I think they are much more useful than working with pairwise comparisons and so on. So just if you have any questions, you can send me, you can reach me on Twitter. I don't know if it's published somewhere else or by email. I can give you directions to the packages and everything to use. So that's all.
Well, so again, thank you, Carlos, for your talk. And we hope to have you with us again in the future to tell us more about your very interesting research.
Another question.
Oh, another question. Okay. Alvaro again says, is the OU model a type of stochastic model? Is it? Could you talk why have you chosen this type of model?
It is an stochastic model. There are, of course, many more, but this is one of the models that is very much used for evolution of quantitative traits in the literature. It has not been applied so much for molecular traits, but for instance, for the evolution of body size or expansion range and these things in ecology, they are very much used. I have used it also because, despite the not being very simple, it's among the simplest stochastic processes that we can work on, and we can actually make inferences. And it was just a first step towards trying to understand better the genetic forces that are contributing to the evolution of splicing rates.
So, if that was the
also maybe one more thing to add in this question is that this model, besides being very simple, it has two parameters that we can interpret relatively easy as selection strength and rate of neutral evolution, which is something that we were looking for to try to better understand what was going on with this splicing rate evolution.
Well, so as I was saying before, we thank you again. We hope to have you again in the future. And we also would like to thank to our assistants for being here and for the insightful questions. And last but not least, thanks to our sponsors, Granada Ciencia, PTS Granada, and MedinBio for supporting us. And in the following days, you could find this video and Carlos' slides in our web, and the link to our web is in the description box below. And lastly, we would like you to complete a form to know if you have enjoyed this activity and hear your hopefully constructive criticism to keep improving our work. You can find the link to this form in the chat. And that's it.
There is a last question, sorry.
Oh, okay. So from Javi Lanillos from CENIO. Hi, Carlos, thanks. For a student in cancer research like me, how are people applying this knowledge in cancer research instead of across species?
I'm not aware of any research applying this type of model to cancer. I think application of evolutionary models to cancer is starting to be quite widespread now, but I don't know if for quantitative traits it has been done. I'm really not aware of that. But it has not been applied a lot to evolution of molecular stuff, so probably not a lot. Okay.
Well, so thank you all for coming. Y como último mensaje en español, os quiero recordar que el próximo martes 23 de febrero, tenemos una sesión informativa sobre formación en bioinformática en España. Súper interesante, que vamos, aquí nuestro amigo Alberto se ha currado un pedazo de documento con toda la información de toda la formación bioinformática en España. Así que si estáis interesados o conocéis a alguien que esté interesado en formarse en bioinformática, no la podéis perder. Os lo recuerdo, el próximo martes 23 de febrero a las 7 de la tarde. Está en todas nuestras redes sociales, en nuestra web también está anunciado y pues será igual, el registro por Eventbrite y bueno, esta actividad pues será en español. ¿Vale? Y nada, pues muchas gracias.
Bueno, muchas gracias a vosotros.